Hello, my BioL 365 class. I believe we left somewhere around this part. So I'm going to go over talking about photosynthetic electron transport chain. So remember we were talking about that this happens on the granium on inside the chloroplast. So on the thylakoid membrane on the granium. So what you see here is the stroma side inside the chloroplast, and this is the lumen side inside one of the granium. It's called electron transport chain because electrons are being shuffled through the series of transmembrane protons. So there is a photosynthetic electron transport chain. One thing that is very important to mention is that the plants are using light or the energy of one photon for multiple purposes. So they use light to split water right here. So you can see there is a protein that is closely associated to photosystem 2 that is helping in splitting the water. But the energy to do that process comes from the light. So the water is the donor of those two electrons, which then go directly into the reaction center of the photosystem 2. Now, already, it kind of happens very fast, so we can say it happens almost simultaneously, that the two electrons will leave the magnesium from the chlorophyll, and then they will immediately be replaced by the two electrons that come from the water. So those two electrons will then go from the reaction center, from the photosystem 2 into the phaeophytin, into the quinone A, quinone B, into the plastoquinone, and then they will go into the iron core of cytochrome complex. And then they will jump the membrane via plastocyanine into the reaction center of the photosystem 1, and then they will be transferred onto the ferredoxin. And we said, remember that electrons cannot just zoom around the cell because they will attach themselves to any chemical that they found first, so they can cause serious damage. So that's why we need something at the end of the electron transport chain that we are going to attach those two electrons. And in this case, it is going to be NADP+. Now, plants are using light in the reaction center for additional purposes, and that is to raise the energy of those two electrons from their low onto their high energy state. And they are doing that because once those electrons are raised onto their high energy state, then they can flow simultaneously, from photosystem 2 via this transmembrane protein onto the iron sulfur core. So I have a table to show you. Sorry, I just gotta fast forward. Oh, where is my table? Sorry about this. I gotta find that table. Sorry about this. So sorry. Oh, right there. There is the table right here. And I don't know why is it so small. Oh man. Sorry guys. Hold on. I gotta find that table. Oh, right. There it is. Okay. So the light is used in the photosystem 2 to raise the energy of those electrons, and then they will flow without any further use of energy onto the photosystem 1, okay? And then once they are raised, they will flow again simultaneously onto the ferredoxin. This is a very important table showing you the arrow which is pointing to the spontaneous flow of electrons. So from here to here, we're going to use energy. From here to here, we are going to the electrons are going to flow without any usage of energy, and then again we are going to use the energy of light to raise them. So we use energy from this point onto this point, and then again from here onto here we do not use energy. So that is very important. Sorry from jumping like this from one slide to another. Okay. Now, these transmembrane proteins, they are seriously multitasking, because one more thing that is happening as well, which is not kind of a kind of picture here, is that once they receive those 2 electrons, photosystem 1 will pump hydrogen protons from the stroma side onto the lumen side. So there will be a lot of hydrogen protons onto the lumen side and less of them onto the stroma side. That is why these proteins need energy to pump the protons from the stroma side onto the lumen side, because they're pumping the protons against their concentration gradient. So there are more of them here and less of hydrogen protons here. And the hydrogen protons try to go back onto the stroma side, but they cannot do that. So every time the electron comes to any of these transmembrane proteins, in addition to them having the 2 electrons, they will pump the 2 hydrogen protons into the lumen side. So in the end of the electron transport chain, here you can see the ATP synthase. So when we talk about respiration, we're going to talk about this transmembrane protein in way more detail. But the only way the hydrogen protons can go back to where they want to be, which is their low concentration, is if they go through this ATP synthase. And as this is happening, the phosphate group is being added to the ADP to create ATP. Okay, so in the light reactions here, you can see we have the NADPH and the ATP. When we talk about respiration, so when we talk about, and I quote, the regular electron transport chain, or the electron transport chain that happens in the mitochondria, then we are going to go talk about this in great details. But for now, let's just talk about the reaction center. So in the reaction center of the photosystem, so you're going to have reaction center which is associated to photosystem 2, and the reaction center which is going to be associated with the photosystem 1. There has to be always a electron donor, which in this case is water, and something that is going to accept those electrons. So that is going to be the molecule of magnesium. So really the antenna chlorophylls are just there to kind of guide the light towards the reaction center. Inside the reaction center, we are seeing 2 chlorophyll molecules, which is called chlorophyll A dimer. So as the light is hitting all those reaction center, antenna chlorophylls are guiding and pointing the light towards the center. So here you can see that it's a very complex structure. So you have a lot of APO enzymes around the actual reaction center. And here you can see water molecules are coming in, they're being split, and this is where, they will leave the electrons. Of course, this happens very fast, 10 to the minus 8th of seconds. So there is a very fast, rapid funneling of energy from water into the reaction center. Now, these are all additional subunits, which, like I said, I'm not going to go talking about into details. The only thing that I am going to mention are those light harvesting complexes, which I say are only functioning and are efficient during low light conditions. So even when there is no direct sunlight, plants can still photosynthesize. And one of the reasons that they can do that is because of, oh yeah, there was the table right here. Let me just jump to the right slide. Okay, so one of the reasons that plants can, photosynthesize and create energy even under low light conditions, it's right. We said the presence of these light harvesting complexes, but also is because photosystem one can kind of happen on its own without the input of electrons from photosystem one. So you will see that there is a protein called proton gradient regulation 5, which can take electrons back to plastoquinone, it's like a loop inside the electron transport chain, and it can run on its own, so that the proteins can keep pumping hydrogens, and the ATP synthase will continue creating ATP even under low light conditions. So this is called non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation. Now, when you see how these transmembrane proteins are arranged, they are arranged in a very specific way that is called lateral heterogeneity. So there are these oppressed regions inside the granule, and then you see the outside non-oppressed regions. So pretty much you will see that the photosystems are compressed more towards the inside, especially the cytochrome and the photosystem 2, while the photosystem 1 and the ATP synthase are located towards the outside of the membrane. And if you remember, I was telling you how the photosynthetic electron transport chain, how the picture was not correct. So this is what is happening. So from the cytochrome, which is right here, the electrons will jump membrane. So it's not going to flow like this. The plastocyanin is going to carry electrons like this. So that's the jumping of the membrane that I was telling you about. So another thing which you can notice right here is look how the molecule of the ATP synthase, how they're located towards the outside. So it would be very inconvenient to kind of trap the ATP inside the granule. So all the ATP that's being created, it's gonna, kind of go outside of the granule into the stroma. So that is why this lateral heterogeneity is extremely important. And just to end up, that cyanobacteria have a very similar complex. However, they have some additional proteins which are kind of, grouped around the photosystem. So there is still an electron transport chain, at least one part of that. They don't have photosystem 1 and photosystem 2 is just one type of the photosystem. However, they have all these additional pigments which are not present in higher plants. So only cyanobacteria possess these. And these complexes are called phycobilisomes, and those additional pigments are allophycocyanin, phycocyanin, and phycoerythrin. So these is what gives the cyanobacteria kind of a purple, dark bluish color, a dark green color. So yeah, I mean they were called blue-green algae at some point. Okay, so that's that. Next slide. Next thing that I'm going to talk about in the next video will be the Kelvin cycle.